The last look of Kitty. Or maybe she'll come for lab this afternoon. Here you go. All right, might as well get started. We have all the usual suspects. So, where are we at? Right, so first up, we have really only one thing to cover this afternoon, and well, this morning, sorry. And we already covered a bunch of it on Thursday. Just get this going. Which is construction and use of transgenics. So we'll finish that up today, but we also have plenty of time to go over any of the material from the last few lectures, right? Because obviously the exam on all that stuff is today. So if you have any questions or want to go through anything, I'm more than happy to do so. Actually, I'll just wait to rub that off in case it's needed again. Speak now or forever hold your peace, as they say. I think that's peace as in peace, rather than peace as in gun. One hopes. Anybody? Well, we can always do this afterwards if it jogs. Going through this work jogs any memories or questions that you may have. All right, so let's get up. Where is that up? Okay, so let's see if I can get the chat back. Yay, there we go. Awesome, worked this time. So essentially it's what we want to do today is just kind of give you some like slightly more formal covering of what transgenic animals are, and also some of the ways that we can ask questions about, you know, why does something work the way it does, or what genes are required for a particular function or process, right? Because in many ways this is it's a fishing expedition, right? So quite often you may have a particular process or phenotype and you've got absolutely no idea what controls it, right? Or what is responsible for it, or what works in the process to, you know, give you the phenotype that you have. And so there are a whole bunch of different ways that you can answer that, those kinds of questions, and we'll talk about a few of them today. But quite often in the process of asking these kinds of questions, you get all kinds of answers that you wouldn't have otherwise expected, right? And so a great example is actually some work that I did a couple of summers ago with a student, which was to look at gene expression over all of the genes in a few different mutant backgrounds, because I expected to find genes which were differentially regulated in one background versus another. And I expected those genes to be involved in metabolism and lipid synthesis, because that's the phenotype that I was working with. And the funny thing was, I didn't find any of those. What I did find was a whole bunch of genes involved in innate immunity and defense against bacteria and worms. And so it's a big like, what the hell does that mean, right? So that's a whole nother direction that I'll probably be going in when I finish up this work. But it just goes to show that you've got no idea what you're going to be finding in some cases, right? And so often the path of scientific discovery, you kind of, when you read papers, it sounds like this very kind of well delineated road that you just travel down, you know, picking up bits of information as you go. And the reality is, oh, sorry, that was my cat sneezing. The reality is that it's actually a very winding road with like dead ends and weird branches that lead off to different places. And that's kind of really where a lot of the skills that you're learning in genetics and a lot of the tools that you would learn to use kind of come in. You know, how do you answer those questions? And obviously transgenics are, it's quite important to define the difference between transgenics and mutants, right? So mutations occur naturally and randomly, and we've been using random mutations for millennia, right? The reason why wheat is, you know, kind of like the height it is and the size of the bushels that it has are the result of, you know, centuries of breeding, right? In fact, even just wheat as it is um, as a plant is a result of potentially thousands of years of breeding. The original, you actually see it on the side of the roads here in San Antonio, which is pretty cool, is, I think it's called teosinte, or is that corn? No, I think that's corn. Getting my plants mixed up. But anyway, they're typically natural uh, predecessors to these various current crops, which were selected for and bred and so on and so forth to get what we see now. Yeah, it's maize. That's definitely teosinte. It was a native of Mexico, basically. Anyway, so there's kind of that, right? So there's just essentially modifying or, yeah, even modifying or using natural modifications to change the genetic makeup of an organism, right? But only using what is already there, right? So you're not adding something from another organism, because if you do, then that's a transgenic. It kind of becomes a little bit fuzzier when we're talking about targeted mutations or genome editing, because then it's, I don't know. I'm not really sure where that fits within that fairly simple definition. It used to be pretty simple, you know, if it has only the DNA that you'd find in that species, however mutated, it's not transgenic. If it has DNA from another organism that scientists have added, then it's transgenic or GMO, right? Genetically modified organism. Genome editing with CRISPR-Cas9 has really kind of blurred that distinction, to be honest, because you can end up with an organism with no exogenous DNA, so DNA from other organisms, but that's not the result of a random mutation. It's a result of a very specific change that scientists added. So I don't know. That's kind of a tricky kind of boundary to define, right? And so this is some cool pictures. I just love looking at fluorescent protein pictures. These are all organisms that are expressing green fluorescent protein, GFP, the protein that I told you about last lecture that I've used and still do. Well, can't right now because my research is shut down, but would use a lot, right? It's a very important tool in my toolbox. And yeah, they do, right? Actually, you can get different colors of mice. So in the jellyfish and corals as well, actually fluoresce. Um, you can get anything ranging from, I think it's cyan is the most blue. You can get GFP, you can get YFP, which is yellow. You can get RFP, which is red. And there are probably some intermediates between those colors based on either naturally occurring fluorophores, right, extracted from nature, or where scientists have kind of tweaked with those proteins to generate different excitation and emission wavelengths. Cats are going crazy. Anyway, so you can actually get yellow mice, you can get green mice, you can get red mice. Supposedly they're very popular in pet shops, although I've never seen any. And it's typically fairly rare with mammals to see whole whole body fluorescence because it's kind of hard to actually get them working that way. But certainly some bacteria, fruit flies, obviously, worms. That worm picture, that's probably a young larvae by the looks of it. And that's almost certainly a cuticle or, shit, I forgot the word of them for a moment, like a muscle or peripheral protein location, right? Because you can see it highlighted all around the periphery, but not on the inside. And so it's really hard to overstate how important a tool that has been. It's really quite, essentially before we had GFP, we would have to actually use things like antibody staining to localize proteins to figure out where they were being expressed, which is really hard and not particularly sensitive, right? And also, you can't do it in living organisms, right? To be able to stain animals, you typically have to kill them, or plants even, right? So being able to kind of look at what's going on in live animals has been a real game changer. So that's just one aspect of transgenics. What I want to do is let's see if there's anything useful here. More here, there's a couple of distinctions to make as well, right? So when we talk about genetic discovery, we typically talk about two approaches, forward and reverse genetics, or forward or reverse genetics. And so forward genetics is really when you start with very little information, right? And so this typically involves mutagenizing the crap out of your organism of choice and looking for mutant phenotypes, right? So it's a fishing expedition, right? You don't know what you're going to find. You might hope to find something in particular, but before you start, you've got no idea what it will show up. And so essentially you mutagenize hundreds of thousands if you've got worms, or maybe hundreds if you have mice or whatever, and you look for a particular phenotype, right? So you screen all of those mutagenized offspring, most of whom die, right? Because being mutagenized is generally not very good for you. But then you screen the survivors and you look for the phenotype that you're interested in, right? So for example, if you're interested in locomotion, you'd look for animals that don't move or that they move funny, right? In worms, those are called UNCs, which stands for uncoordinated. And those genes could be involved in a whole range of different processes, right? You've got no idea when you start. You're simply looking for the phenotype. And then once you have a phenotype, obviously you try and get rid of all the other mutations that are in that organism. And then you start trying to figure out what that gene is, right? And elite for UNCs, for example, you have mutations in body wall muscle, you have mutations in neurons, you have mutations in things that people don't even know what they're involved in, right? And so you just get this giant pool and you just basically work through it and figure out what all these different things are, what the genes are and what those genes are involved in. So that's very much like the kind of the old school way, which still definitely has its place without doubt. But now we've got a whole bunch of other ways of figuring out stuff, right? And this is known collectively as reverse genetics. So in this case, typically, you start with something known, right? So you might know what the gene is. It might be, okay, we've got this particular list of genes, right? We're going to selectively knock them all out, or we're going to overexpress them all, or whatever, right? And then you look for what phenotype that causes, and that tells you essentially what its role is in that process. So that could be by targeted mutagenesis, right? Which we'll talk about in a little bit, which is a fairly new discovery technique, right? Which is in humans and flies and stuff, they've had kind of that sort of technology for a while, but worms certainly haven't. It could be simply starting with a list of genes, right, and knocking them down using RNA interference. So, for example, work that I did back in Penn State, I was looking for essentially genes downstream of a cell signaling receptor, right? And so the idea there is you have a what's called a sensitized background. So in this case, a mutant phenotype, right? So what you're looking for is anything that changes that mutant phenotype back to normal, or at least normal within the context of whatever you're working on. And if you find something that does, then potentially that gene that you've knocked down is downstream, right? So this is a kind of a practical application of epistasis, right? Which we talked way back when we talked about Mendelian genetics, right? And so for that particular project, I had a list of 257 kinases, right? So these are proteins that add phosphate groups to other proteins, either to activate them or inactivate them, right? So a post-translational modification. And basically just started working through those. I think I got through like 100 or 120 of them before I focused on characterizing the candidates. And so anyway, I already started with something known, right? I didn't know which ones of those would be responsible for my particular phenotype, but I already knew what the genes were that I was messing around with, right? So that obviously takes out a lot of the guesswork and the legwork because you already know what you're working with. And that's very much called reverse genetics. And there are lots of different ways that we can do this, right? So essentially, there are a few different things that we want to do. One, we want to be able to get that change into an organism, right? So typically this is called germline transformation. We'll go over that in a second, right? Because we don't want to just look at that modification or change in that individual. We want that to be heritable, right? So we want it to be in every single offspring, ideally, of that individual so we can study it, right? We also need to be able to target a mutation, right? We're going to talk about that down here, right? Because otherwise we're basically back to square one with forward genetics. Pardon me. And ideally, this is where the latest hotness comes in. We want to have control over that modification, right? And this is really where CRISPR-Cas9 comes in. And this, I don't know, I'd have to double check. I'm not sure if they got the Nobel Prize for medicine, the people who discovered this. This was discovered, I think, in 2009 or 2010. So this is very new. In fact, it's so new and it's such a huge potential that the two groups that discovered this are engaged in essentially a multi-billion-dollar lawsuit to try and figure out who really discovered it first, right? Because there's that much money involved in this particular process. It really is it's another one of those step changes. You know, we talk about things which kind of like totally change our understanding. So, you know, Mendelian genetics, Thomas Hunt Morgan's linkage work in flies, structure of DNA, right? Invention of PCR, and the discovery of thermophiles in Yellowstone, right? So being able to do PCR on a big scale. What's the other ones? GFP and RNAi, so RNA interference, being able to knock down genes without permanently changing them, right? And being able to visualize expression in vivo. And basically this is the next one. So this is another one of those changes which will change the face of science and most likely society as well, right? Because those two things are very much linked together, right? You can't really separate one from the other. And so an example of how much that has changed society, I don't know if any of you read about the Chinese researcher, I think it was 2 years ago, who genetically modified two female embryos, essentially, who are now girls, to be able to be resistant to HIV, right? And so those changes are permanent, right? Every single cell in those two girls has that change, and that change will be transmitted to their offspring and probably to their offspring as well, should they choose to have children, right? So we're now capable of, right? It's heavily frowned upon, thankfully, but we're now capable of actually genetically modifying humans. So all of those sci-fi films like Gattaca, what else? Not Minority Report, but there's a whole bunch of different sci-fi films that kind of talk about the rise of human modification and eugenics, and a bifurcation of society based on those who can afford it and those who can't. Well, this is actually the like ground zero for all of those science fiction. Yeah, I haven't watched that. I need to watch that with the kids. But this isn't science fiction anymore, right? Not with the kids? Okay, I'll watch it with my wife then. My kids are pretty robust. They like horror films more than I do, actually. And so in the next 10, 20 years, kind of keep your eyes and ears open, and you may hear of this becoming more common, right? Because this is like Pandora's box. Once you open it, you can't put that technology back inside. The only question is how that technology is going to get used. So who knows, right? There may be places in the world where you can go and get your future offspring genetically edited, right, to have traits that you wish, right? Get rid of disease, one hopes. Perhaps start even like in Gattaca, if everybody watched that, which is a really cool film, trying to get rid of undesirable personality traits, right? You want your kid to be good at sports? Okay, well, let's edit those protein producing genes and kind of neural development genes, right, which allow kids to build muscle faster and respond faster. So you could get like totally ripped, like 12 and 13-year-olds that have like six packs and stuff. I mean, it's not science fiction, right? It's kind of the present, which is really scary. Okay, so essentially, we need to get this, whatever modification we make, we need to get this into the germline, right? And so typically, it depends on the organism, right? So with worms, for example, we simply inject this DNA into the ovaries or the uteri, because it has two, of the worms, and then some of that DNA gets incorporated into offspring. In others, we basically inject that DNA into one of the nuclei. This is with higher order animals like mice. And basically that then, one hopes, results in that DNA being incorporated into germ cell nuclei, right? So there's a good picture down here, right, which kind of illustrates the issue. So basically what happens once you do that is you get what's called a mosaic, right? Because obviously, let's go back up here. If you inject it into one nucleus or one pronuclei, let's say, or even if you inject it into the nucleus of an oocyte, for example, of an egg, right? There's no guarantee that that DNA is going to get shared equally between all offspring at each cell division, right? And even more so if you do it this approach into one of the two pronuclei. Pardon me. Because essentially only the offspring of that one, what will become that one cell, right? Once cell division occurs after fertilization, only the offspring of that one cell will get that DNA. And so what you end up with is basically a patchwork, right? Where you'll get, and if we kind of zoom in a little bit, it's kind of illustrated quite nicely here. You'll get offspring which either may not have it at all, right? Because, you know, it didn't end up in that particular lineage, or you'll get offspring which have, you know, in different places, right? And so then you breed that transgenic mouse or modified mouse with an unmodified mouse, and you select and you hope that that DNA got into its germ cells. And if it did, then all of the offspring from that mating will be transgenic, right? So whatever you do, you'll almost certainly end up with a mosaic issue, right? Or mosaicism, where not all of the cells of that individual have that piece of DNA, right? That modification. And so you need to go through a few rounds of breeding until you get one where it is the case. And that's often actually where things like these markers are useful, right? So it's very hard if, you know, let's say you're, hang on, let's look at this one. This is always a fun picture. This is an obese mouse, kind of sad, funny as well, and kind of, you know, it's kind of in between the two. But it's not always possible to see the phenotype visually of whatever modification you made, right? So quite often you need to use some kind of marker, right, to tell you that that animal has that modification, right? And so GFP is often one of those. So you can tell that these three glowing mice pups are transgenic, and these three aren't. So these three will probably go to feed in snakes or something like that, and these three will get bred again when they're old enough to try and kind of make that that lineage permanent. Those will become mousicles somewhere. So anyway, that's the general process, right? We need to get that DNA into the germline of an organism because then it will become heritable. If it's in the somatic cell line, it will affect that organism potentially, but it won't be transmitted to its offspring. Cough it up. Cat's got a furball. Right, so that can be simply a reporter piece of exogenous DNA. Right, and by exogenous, I mean not normally found in that organism. Right, which might be I don't know, antibiotic resistance or some kind of protein that you want to express, like insulin and yeast, for example. It could be a mutant copy of that, of a gene of interest, right? Or something where it's overexpressed, you know, something that is not normal, right, in that organism. But really what you want to be able to do, and this is where the real power of genetics comes in, is that you want to be able to make a targeted change to that organism. Say you suspect that gene X is really important in a particular process, right? So you could, by forward genetics, by mutagenesis, random mutagenesis, try and find mutations in that gene, but you have absolutely no control over whether or not that happens, or even if you do get mutations, what kind of mutations they are. right? They could be insertions, deletions, they could be missense, they could be nonsense, right? So it's really a box of chocolates when it comes to the outcome of that. So what you really want to do is be able to do a targeted mutation, right? And that's where gene targeting comes in, right? And so this is kind of slightly old tech. We'll talk about like the new hotness in a little bit. But the idea here is that essentially you have, let's make this a little bit smaller, right? You have a gene of interest, right? So this is your target here. And the old school way of doing it was to basically create a vector, right? Which is essentially a carrier of your gene of your DNA of interest, right? To get it into the organism. And you create it such that it has regions which are identical to the bits of DNA either side of your gene of interest, right? Because then, and so that could be a little bit of DNA in the middle, or it could be something that replaces the gene entirely. So that when you essentially use homologous recombination, and typically that's via enzymes that you add yourself, right? Then you may get homologous recombination where either the target gene will be interrupted, right? So you'll get a complete loss of function. Or you could replace that piece of DNA or that target gene with a piece of DNA you created yourself, right? And so this may be a full copy of that target gene, but with like one base different, right? So one, like a point mutation that you're particularly interested in. Say you want to look at the role of a particular active site in an enzyme that has a particular conformation, right? And it has to be that shape to work. And part of that conformation comes from, say, this amino acid here. Say it's got a positively charged residue. So you want to see how important is that amino acid to the function of that protein, right? Without getting rid of the protein completely. Then you can change the codon that encodes that amino acid and change it to something that, say, is neutral, right? Doesn't have a charge. And then you can see, well, what effect does this have on protein function? And the thing is, you've got to replace the target DNA with that new version, right? You can't just supply it like on the side because you've got to get rid of the original function, right? You can't have those two things side by side. Otherwise, you may never see the effect of your particular modification. And so typically you do that in stem cells or you do that in embryos, right? So once you've done that, then obviously you stick it back and you can end up with something that looks a bit like this, right? So this is, as the title says, it's a leptin knockout. And leptin is pardon me, a hormone that you produce when your stomach is full. And basically it tells you that you are full, right? So like the classic dieting advice is eat slowly, right? So that your body has time to realize when your stomach is full. If you eat like really, really quickly, you can, overeat essentially before that mechanism kicks in. And so this mouse doesn't have that, right? So it always thinks that it's hungry. Well, it is always hungry, right? Because it always thinks that its stomach is empty. And so it overeats, and then you get this obese mouse. So there you go. Maybe it needs its stomach stapling or something. Okay, so that's all well and good. Oh, let's talk a little bit about RNA interference first. So sometimes you don't want to do that permanently, right? So in this case, this animal, it survives to adulthood, right? Might be a little bit on the chunky side. but it's not a gene which results in death of that organism or kind of developmental issues that prevent it from getting to reproductive age. And so there are an awful lot of genes which need to function for that organism to be functional, right? And so you can't knock those genes out, at least not very easily, right? Because if you knock them out, then that organism won't survive, right? It won't make it to adulthood, and you're essentially hitting a dead end, right? You might be able to mutate it, but then you can't get any offspring from that mutant. And so a very powerful technique, which is kind of like the other half of my toolbox, more or less in my lab, is using something called RNA interference, right? So this isn't a knockout, right? You don't change the genetic information of the organism that you're studying. What you do is you use, let's see if we can go back. Where is it at? Here we go. Let's see. Hopefully you can all see that now. Essentially, you use an existing system, right? We talked about this when we went over it. Using these small interfering RNAs over on this side, right? And so now, instead of this being an endogenous process, now you supply that double-stranded RNA, right? But now what you do is you basically create that double-stranded RNA corresponding to your gene of interest. Let's say you wanted to knock down leptin, right? But you don't want to have a mutant mouse. Then you can either inject or feed worms in my case on bacteria expressing this. You can inject this double-stranded RNA into cells. You can even soak cells in double-stranded RNA, believe it or not, and those cells will take that up. I don't know why, but they do. And then what happens is it triggers this whole mechanism, right? So typically they're around a few hundred base pairs long, these double-stranded RNAs that you produce. They get cut down to 22 to 25 nucleotides by this enzyme called DICER, which is, again, part of your cell machinery. And then this RNA-induced silencing complex basically gets rid of one strand and then scans the entire transcriptome, right? All of the transcripts that are currently being produced in that cell. And where it finds a match, then that mRNA is degraded, it's cleaved, and it's then open for degradation, right? And so this is an astonishingly powerful technique to look at gene function, right? Because you don't have to go through all that laborious germline transformation and crosses and screening and things to get a mutant. And also, you can do it at particular times of an organism's life that sidesteps its requirement for some other process, right? So if you, for example, want to look at the role of leptin, right, in adult mice, right, you could either insert a conditional knockout, which is only activated at a certain point, like by a chemical in its food, or you can use RNA interference to knock it out only in adults, or not knock it out, knock it down, sorry. And so it gives you an awful lot of control. And as I talked about earlier, I said I started with a list of 257 kinases, right, to screen. I've still got 150 odd in a freezer back in Pennsylvania. But if you want, you can do the whole genome of C. elegans, right? I think it's 19,500 genes have an RNAi clone in a bacteria that you can pick out of a library in a freezer and feed it to worms and then see what effect that knocking down that gene has on whatever it is that you're interested in, right? So that's allowed an awful lot of, essentially it's kind of like forward genetics, but reverse genetics at the same time, which is particularly neat, I think, and extremely powerful as well. And then you can even use that with these GFP worms, and you can just knock down thousands of genes, and then you actually can put these worms through what's called a worm sorter, which is basically it's like a cell sorter or a FACS, or FACS, if anybody's ever heard of one of those. It's essentially a machine which you pour a whole bunch of cells or worms in the top, and then it sorts them based on whatever criteria you specify into different tubes, maybe different sizes, maybe different levels of fluorescence. It could be, a bunch of different things. But there have been experiments that I've read the papers for where they look at the effect of this knockdown on fluorescence, right? So they just knock down thousands upon thousands, tens of thousands, literally, of genes, pardon me, at once, and then use this kind of machine to take, tens of thousands of pictures of those worms, and then they use computer algorithms to figure out what's going on. Right, so now instead of just working on one gene that you find through a mutant screen, you can work on tens of thousands, right? So you can really take all of that genomic information, right, all of that sequence information, and actually start figuring out what it means, like what is it involved in, which is really neat. Does anybody have any questions so far? There's a lot of stuff to absorb. Cool. So at least in worms, and definitely in even though these genome editing techniques were available in flies and mice and other model organisms for a while, probably like last 20 years or so, they were very clunky and hard work, right? Extremely so, in fact. And working with mice is generally a pain in the ass anyway, because they're expensive and they have a long generation time. So there wasn't really kind of an easy way of doing a targeted modification until CRISPR-Cas9 came along. And so this was discovered originally in bacteria. Actually, I have a friend from way back who works on this in bacteria, not like the genome editing or modification, but actually the system itself in bacteria. And essentially, this is a form of immune memory for bacteria. So you know how we have B cells and antibodies and stuff kind of floating around in our body, which have a memory of pathogens that have infected us over the course of our life, right? And some of those last for longer and some of them less so. But if we've been infected by, say, measles, or we've had a measles vaccination, right, we have a memory of that. And so CRISPR-Cas9 is really a bacterium's memory of what viruses it or its parents or grandparents or whatever have been infected by. And so essentially what it does is it keeps like little snippets. So this is, I can't remember what CRISPR stands for, but I know PR stands for palindromic repeats. Can't remember all the rest of it. You can look it up. But essentially they're little snippets of the viral genome, right? And it just tucks it into a bit of its genome, right? And so it has this whole series of little virus chunks, right? Not that those virus chunks will be able to do anything, right? They're non-functional, but they're like like when you go to a national park and you get a fridge magnet, right? They're essentially like a little memento of that viral infection, whenever that happened. And so every time the bacterium is infected by a virus, of which there are, thousands of them in nature, it produces a copy of all of those repeats. And basically it uses that and it kind of scans that incoming DNA or RNA for matches. And if it finds a match, then the Cas9 protein cuts that DNA or that RNA. so that it's no longer functional or get degraded, and that bacterium won't get infected by that virus, right? So it's really like an immune memory for that bacterium. And it's made up, it's kind of like a proofreader and pair of scissors at the same time, right? So it kind of scans through all of the exogenous DNA, right? Because obviously it doesn't cut its own, because that would be a disaster. But anything new coming in, it scans it, and if it finds a match, it cuts it. And those cut ends are then essentially signals for nucleases. Yeah. So obviously, if the bacterium hasn't been infected by that virus, it doesn't do any good. But if it survives that infection, it will make take a copy of a little bit of that viral genome, and it will stick it into its series of repeats for the next time, right? And so this is a kind of a complex process to explain, but essentially there are a varying number of pieces, right, which come together to produce a particular outcome, right? And the two main important pieces, oops, go back down here. Let's go see if I can scroll up a little bit. There we go. This Cas9 protein, right, which is essentially the bit that does stuff, right? So this is the protein that cuts the DNA, and what's called a guide RNA. And so this guide RNA is the bit of nucleic material which will match your target, right? So in bacteria, this is essentially what corresponds to that fragment of virus. In our hands as scientists, you create that guide RNA, right? So you specify, I want this bit of DNA to be turned into a guide RNA, and this bit of DNA corresponds to whatever gene that you want to modify, or part of the gene you want to modify, right? And so when you supply that guide RNA and that Cas9 protein together, typically you inject the two of them into a cell, right? Then that guide RNA there has to be a PAM, and this is like a frequently occurring sequence. It just needs to be nearby, essentially. That guide RNA and the Cas9 protein kind of form a complex, right, called the effector complex. Pardon me. And they will go around and they'll unwind DNA, and they'll scan along the genome until that guide RNA matches a particular sequence of DNA, right? Which is what you see here, right? From, standard just base pair complementarity. And it has to be it's not super exact, right? So there are downsides to this technique, but it's pretty accurate. As soon as that match is found, the Cas9 protein cuts the DNA, causes a double-stranded break, right? Which, as I've told you in the past, is a pretty catastrophic event for a cell. Typically, if you have a double-stranded break, which is normally caused by things like ionizing radiation, like gamma radiation, you have to fix it, otherwise the cell will die. And so how that's fixed depends a lot on what you do. So this is where the potential extra component comes in, and this is really where a lot of the power of CRISPR-Cas9 comes in, right? So originally, like before this was refined as a technique, that was it, right? So your guide RNA targets the Cas9 to your target sequence. Cas9 causes a double-stranded break in that DNA, right? So it cuts both strands. And then the cell, it basically kicks off a emergency DNA repair mechanism called non-homologous end joining. And really what that means is it's like you got two bits of string, let's just stick them back together again and hope, more or less, that they're the right ends to stick back together. And so that's very much a last ditch, oh crap, we're going to die kind of repair mechanism. And as such, it's not very good. It will stick those two ends back together, but quite often it results in either bits of DNA getting duplicated, bits of DNA getting deleted, or even little bits of DNA getting inserted into that repaired join. Right, it's kind of like using glue to glue things back together again, right? It doesn't look quite the same as it did to begin with. And so those small changes, even with one or two base pairs, right? It doesn't have to be much, basically screws up that gene, right? So really the purpose is to force this non-homologous end joining repair mechanism into action, which we know is not especially faithful, right? It's not very accurate. And that lack of fidelity or that inaccuracy causes a mutation, and so you get a knockout, right? It could be a frame shift, so you get like gibberish as a protein. Could result in a stop codon emerging, so it's going to be a truncated protein. Whatever it is, that protein is no longer going to function, right? So this is like the classical CRISPR-Cas9 kind of approach, right? This was how it started off, and that is not trivial at all, right? We literally could not do this in C. elegans before this technique came along. So if you wanted to mutagenize a particular gene, you just had to do a shit ton of mutagenesis and hope that a mutation occurred in your gene of interest, just by random chance. Right now, you, or if you've got a buddy that knows how to do it, because I don't know how to do this, but I have a friend who does, you design this guide RNA to that gene of interest, get it into your cells or into the germ cells, and you'll get a mutation or knockout of that gene, right? And so, can this be used to stop cancer growth? Ooh, that's an interesting question. It's a very difficult question to answer because cancer is you, right? So it's not different from you. It is different from you in some ways. But potentially, yes. If you had a mechanism of getting this whole system, right, you'd need to get both the guide RNA and the Cas9 protein expressed in those cancer cells. If you could do that, say, via a modified virus, right, which only infects cancer cells, and I think they've been trying to work on those. Well, yeah, you'd have to know what gene was causing that cancer or was responsible for that. They haven't tried that yet. I don't think so, right? Because there's a lot more, you'd have to get a lot more ducks in the air to be able to get it to work. One, you'd have to get a virus which recognizes that cancer cell only, right? Because you really don't want to screw this up in non-cancer cells. Two, you'd need to, hang on a sec, Ali. You need to modify that virus so it express both the guide RNA and the Cas9 protein. And then those two things would have to be able to heritably knock out that particular gene. So in theory, it is possible. I haven't heard of anybody using modified HIV because that's kind of terrifying. Typically they use things like varicella viruses, so large DNA viruses that have a big enough genome to be able to mess around with them to get this stuff into them. I know that there are differences in cancer cells which identify them as cancer cells to your immune response. So immunotherapy is a really big thing right now, where you basically, yeah, well, that's a good field to get into. Basically, you raise antibodies to cell surface proteins of cancer cells, and then inject those into the patient. So it's very much a patient-specific treatment, and essentially trick your immune response or your immune cells into destroying those cancer cells specifically. So immunotherapy is a very big deal. I don't think, I know there have been some trials of, I think they use it for congenital diseases such as like ALS. I think there's trials going on right now for sickle cell disease, and maybe a couple of others where essentially they're switching in a functional copy of a protein. In sickle cell disease, it's actually a version of hemoglobin that you find only in children, or in neonates, actually, in babies. And that seems to be quite effective. I don't think it uses this technology, though. I think it's using like an older version of recombinant DNA, homologous recombination technology. So it's definitely possible, right? But this is like, these are clinical trials that are going on right now, to try, because essentially to modify an adult, modifying an unborn child is in some ways somewhat trivial, because you just have to start with a germ cell or some stem cells or something like that, right? So, and then that gets spread amongst all the cells, one hopes, or most of the cells of that individual through the process of development. Actually changing DNA in an adult or in a grown child, pardon me, is really hard, because you have to then get that modification into every cell that requires it. So yeah, not trivial at all. But yeah, certainly in terms of cancer treatment, things have changed massively in the last, just in the last 5 years, actually, in terms of treatment. So it's not, there are obviously, you still have you know, blanket treatment like chemotherapy and radiation therapy and things like that. But now we're able to kind of target that therapy better, basically through genome sequencing. So we know which, there are certain individuals are refractive to chemotherapy, but certain chemotherapies, because they have a particular mutation or allele for a particular gene, right, that might metabolize that drug. Or their cancer cells may be more prone to developing resistance, right? So the first step is to figure out what exactly type of cancer do you have, what are the genetic changes, and then modify treatments accordingly. The next step is tailoring that treatment via immunotherapy to that one individual, right? So this isn't just kind of, hey, we've got a population of 100,000 people that have cancer. This treatment works for these 2,000 people and not these 98,000, right? Now it's actually taking cells out of an individual and creating a targeted treatment for that one individual. And so the next step is essentially what you're describing, are these essentially genetically modifying cancer cells to be able to turn them off, right? Whatever might be causing them to be dividing uncontrollably, if one can figure out what that is, and we have a pretty good knowledge of how this works, not perfect, but a pretty good knowledge, then potentially you could make those cancers go inert, right? And so once you've done that, then you simply cut them out via conventional surgery, and then one hopes, will not get a resurgence of that cancer. Well, all biology is interesting, right? Cancer biology is, I don't know, I've always had a like passing interest, but whenever I get more into the details, it becomes a bit too mechanistic for my liking. So if you like kind of knowing how all the different details kind of work together at a very fine grain level, cancer is an excellent field. It's kind of like a great field for people who don't like people enough to be doctors, in a sense, right? You really, really like those details, and you like how all these different things fit together. And you really like zooming in. If you like kind of like the bigger picture stuff, then cancer is probably not the field for you. Like,
videos I've done in the description of this
yeah,
one.
large,
As the system's full of air right now anyway.
not necessarily
Nice, way
ecology.
better. I'm
Hey,
using
someone has
compressed
their
air,
microphone
so that's what I'm doing right now.
unmuted. I don't know who that was. Thank you,
Remember
Austin.
the best way to add air? I used to have a
Hey,
better.
Austin, mute your mic. Thank you. Um, yeah, if you like yeah, don't worry about it. It's okay. If you like kind of like the bigger picture, how do things join together at more of a systems level, then other aspects of biology may be more interesting. Kind of like developmental biology kind of is essentially between the specifics of cancer and like the big picture ecology and evolution type stuff. Yeah, so to be honest, um, if you like medical fields and you like working on medical related stuff, then one thing that you either kind of going into lab research, lab technician, working in a pharmaceutical lab and working up from there is one aspect. Or if you really want to shoot for the stars, and this is not a trivial deal, you can get what's called an MD PhD, or is it an DPhil? I can't remember what the precise term is, but essentially it's an MD program where you also do a PhD at the same time. So it's like 8 years or something crazy. Yeah, yeah. Well, there's that aspect to it as well. Um, but that's another potential route. Those are extraordinarily competitive, right? Not trivial whatsoever to get into those. But that essentially gives you both worlds, both the medical aspect and the research aspect at the same time. So those are um, that's another potential option to explore. So anyway, the thing that really changed everything, right, in terms of genetic engineering was this side, right, this bit over here. And so essentially, you have these two components, right? You have your guide RNA to target Cas9 to your gene of interest, and you have your Cas9 protein. But now you also have, where's a good picture? Keep that. There we go. You also have a vector with DNA that matches your gene of interest, right? Particularly either side of it, right? And now what you can do is you have control over the bit of DNA in the middle. So you can do a lot of different things here. You can just use this as a basic targeted interruption, right? You can stick a GFP gene in here, right? So now you insert a fluorescent reporter into that gene, or maybe just behind the promoter, or maybe in frame with the actual protein itself. So you end up with a hybrid or chimeric protein. Or what you can do is you can basically recreate that segment of DNA that you're going to be swapping out. So it's just the same as the target gene, except for the change that you want to make. So if, like I was saying before, you want to change out one amino acid, right? The old school way, or the original way of CRISPR-Cas9, just doing this duplication or deletion, is way too random, right? As to what change you're going to get. So now what you do is, at the same time as you provide these two components, you also provide, typically it's a vector or plasmid, right? So it's a circular piece of DNA, maybe, it could be linear, right? But whatever it is, it has two regions which are homologous, the exact matches to your gene of interest, to your target. And so now what happens is when you create this double-stranded break, now when the cell's like, oh shit, right? This is bad. Now it scouts around and says, oh, I've got a matching bit of DNA that I could use to fix this, right? So if you cast your minds back to homologous recombination as a DNA repair mechanism, right? This we covered a while back, I can't remember when. This typically happens when a cell is dividing, right? And you have those kind of sister chromatids present that you can use as templates, right, for those breaks. But now it doesn't have to be the case because you're providing that little bit of homologous DNA yourself, right? So this is something that you add in there. And so basically when you create that double-stranded break, right? This isn't a great image, but it will serve the purpose. The cell repair mechanisms will kick in, and it will go through the homologous recombination repair side. And what that then does is it will swap out the broken bit, right? The bit that you've created the break in, and it will swap in your modified piece of DNA. And so what you end up then is either a new bit of DNA inserted, right, which might code for something that you want to be there, say a reporter or whatever, or it might simply change the sequence of that target gene in ways which you decide, right, depending on, you know, what you're interested in, right? So another application, for example, is it ALS? Yeah, I think it's ALS, is caused by a trinucleotide repeat, right? So extra copies of a, I think it's glutamate, or no, glutamine, that's it. Results in multiple glutamines, which then ends up with, oh shoot, what's the word for it? Aggregates, right? The protein forms an aggregate and then that cell dies, typically a neuron. Right, so now what you can do, for example, let's say you want to figure out what's the dividing line between not having this disease and starting to show symptoms and being really severe, right? Rather than waiting for that to occur naturally, now you can just swap in, you know, different amounts of those trinucleotide repeats, and you can just look at the offspring and say, that mouse is fine, that mouse is going all over the place, right? You can actually figure out exactly where that shift occurs without having to rely on natural processes to generate that particular number of repeats or that particular genetic context, right? So really, this is now a matter of what can you imagine, right? It's not a matter of, hey, I want to do this, but I don't have the tools to do so. Now we actually have the tools to do this. And this is actually something that is spreading amongst all kinds of organisms, right? Organisms that typically were really refractory to genetic modification, like really hard to work with. It's being used to modify crops, right? So an awful lot of kind of economically important crops or plants like rice, wheat, maize, rather than going the old GMO route where you like slap some bit of foreign DNA in there, which tends to worry people a lot, right? Because they worry about off-target effects. Now you can actually modify existing DNA, existing genetic material, but without adding anything novel or new in there, right? So really you're just, rather than waiting for natural mutation to give you something that you want, now you just create what you want, but you're not really adding anything new. You're simply working with what is already there, right? So it's a way of making kind of genetically modified organisms without all of the things that scare people about genetically modified organisms, right? And so really you're just speeding up natural and artificial selection to get what you think you want. Whether it turns out to be right or not is another issue. So just to finish up, this all may sound like super rosy. Always have to be a bit of a Debbie Downer on this, but there are also problems with this, right? One is that this isn't 100% accurate, right? So you can get off-target effects. You can actually, minimize that. You can use there are modifications to the system where you use 2 guide RNAs rather than 1. So it's more accurate because the likelihood of both of those ending up in the wrong place and together is very slim. You can also do like genetic screening as well, make sure that you didn't get an off-target effect. But the biggest issue is in humans, right? So the issue here is that we don't actually really truly know what we're doing, right? And once we make this change, it's permanent, right? And it's not just permanent for that one individual. So say you had a particular disease, right? And we modified you, or maybe you had a particular disease and your child, you didn't want your child to have it, right? So you had your oocytes or sperm genetically modified, or even your zygotes, right? Your fertilized eggs, genetically modified to not have that that issue, right? That mutation, whatever it might be. Problem is that change, whatever that change is going to be in every single cell of that offspring, and it's going to be passed to that offspring in turn, right? So your offspring didn't get any say in having that change made for them, right? You made that change, essentially yourself for their benefit. And they certainly don't have any say in the fact that that change is going to get transmitted to their offspring, right? And so the case of the Chinese researcher in China is a really interesting illustration, right? Because the modification that he did was to a T cell, so an immune cell surface receptor, right, which facilitates the entry of HIV into those cells. And so he made that change. Obviously, those 2 girls didn't get any say in that. And yeah, but that's true. But you also don't get any say in what gets handed down to them either, right? Essentially, they get what they get, and it's totally random, right? So the issue in the Chinese case is that we don't actually know truly what that receptor does, and it may actually end up making them more susceptible to other viruses, right? Which are it's kind of like common cold or whatever, right? So we don't actually really know really what will happen once that child is an adult, right? We don't know enough about human genetics really to make those kinds of changes. And the problem is that once that change is made, that's done, right? That individual will have that change, and every one of his offspring will have one copy of that change, and its grandchildren have a 50% chance of having that change. right? So I don't know. I mean, that's the funny thing, right? That's where our lack of knowledge comes in, right? So, um, you know, if you think about what makes an athlete successful, right? Now it is, you know, a random combination of alleles which make you faster or stronger or whatever. And there's also a bunch of other stuff in terms of, you know, motivation, tolerance to pain, you know, willingness to take drugs, whatever, right? All of those different things. And so if you think about all of those different phenotypes, right? Like what you see, like the consequences, how many of those are going to be controlled by multiple genes, right? And how many of the genes that control those phenotypes control other phenotypes, right? So if you make an individual, like an offspring, more single-minded, more tolerant of pain, and more willing to take drugs, right? Well, maybe you get a big dumb athlete. Maybe you get like a really angry drug taker. You know, you just don't know, right? And so, because so many things that make up us as humans are controlled by many different genes, and many of those we don't actually know what genes control that. How do you know that whatever changes you're making will actually give you what you want and not something that you don't want, right? If you try and make someone more uh, intelligent, right? You know, try and edit your embryo so that your child becomes like super brainy, right? Because you want them to be the next, I don't know, Bill Gates or Mark Zuckerberg. Actually, I wouldn't want my kids to be either of those two. But say you did, then you might end up actually getting, you know, a sociopath, right? Because the same traits that lead to people to be successful in those kinds of situations in other people lead them into other directions, right? Which are less favorable, right? So that's just, you know, kind of like a worm in your ear, something to think about. It's an interesting topic to talk about, right? Particularly now that we have the means to take away some of that randomness, right? So you can actually, even now, if you go through IVF, select for children or select for eggs rather that do not have particular diseases, right? So you can screen for Down syndrome, you can screen for um, other obvious genetic abnormalities, right? So it's the technology is there and the systems are already in place, and now we actually have the technology to not just screen for obvious things that we already know of, but also to start modifying traits. You should all watch Gattaca. It's an old film, but it's a really cool one. Very stylish too. Really cool cinematography. Um, but yeah, and also don't forget too. I mean, I don't know if you know how expensive IVF is, but if you do it privately, it's tens of thousands of dollars, right? Yeah. Oh, cool. Yeah, I mean, Gattaca is like one of my favorite science films. It really is. But also, don't forget that this stuff isn't cheap. So it's not just that all people will have this available to them. You know, it's really only going to be the people that can afford it that can that will do it. And so, you know, what are the societal consequences of that, right? If only the offspring of rich people are super intelligent or fabulous athletes or live to 150 or whatever, right? Or any of those kind of desired traits. And people in the lower socioeconomic echelons can't afford it. You'll end up with that bifurcation of society into, even more so than it already is, into people that are super and the people that are just normal. So yeah, it's really, really interesting. So anyway, so that's pretty much all I've got for this. Anybody have any questions? Questions. It's a cool discussion. Bioethics is actually a really neat topic to talk about. You know, particularly when you have the means to do something, and it's then about whether you should do it, not whether you can do it. And Gattaca is, I think, was that late 90s? Had a very youthful Ethan Hawke in it, that's for sure. I think late 90s, early 2000s, something like that. You know, and so it wasn't that long after that was made that the means became available to do what they were talking about. Transcriptional regulation, for sure I can. Tell you what, why don't we take a few minutes break, because people are probably a little bit brain bashed. I know I am. And hit back here at 12:30, just so you can go stretch your legs and the like, and then we'll go over those questions. Okay, how's that sound? Well, I'm going to assume that's an affirmative. So let's meet back here in 10 minutes, and then we'll go over any questions about topics. Okay. Let's pick up. So I'm assuming, Angela, that you're talking about gene expression in eukaryotes, correct? Or do you want to touch on prokaryotes as well? I don't mind. It's really up to you. Angela, are you there? No, I think she's there. Yeah, she's there somewhere. Okay, so let's do prokaryotes first. Crab sticks, come on. Where's that one gone? Here we go. Okay, so what aspects? Let's narrow it down. What things are causing you issues? Lac-operon, trip-operon, coordinated transcription and translation, catabolite activator protein. What do you want to cover? You need to give me some pointers. And don't worry about like speaking up. I'm sure whatever questions you have will help other people too. Come on. I'm raising my eyebrows. All right, let's turn it around. Let's give you a little mini quiz. So when it comes to what will shut down the trip-operon completely? Oh, that's okay, Andrea. What shuts down a trip-operon completely from being on all the time to being completely off? Trip repressor and what? Tryptophan, right. So what do we call the TRPR protein in the absence of tryptophan? It has a particular name. It's not just a repressor, right? Because that's what LAC-I is. What kind of repressor is it? Yeah, that's right. So aporepressors are repressors that only bind DNA in the presence of their co-repressor. So if TRPR is the aporepressor, what's the co-repressor? You already told me what it is. Yeah, tryptophan, right? So that's a really big difference between the lac operon and the trip operon, right? So the lac operon, lac I, will bind to the lac O operator in the absence of lactose, right? So lactose or allolactose is the inducer because it causes that protein to leave. In the trip operon, it's the other way around, right? That repressor will only bind when tryptophan is present at high enough levels, right? So you got to have that difference kind of clear in your head. All right, there's another question. Is the trip operon repressible or inducible? Correct, Peyton. It's repressible because normally it is on, right? So it is negatively regulated and repressible, right? Because that's saying that it is normally on and it will be turned off in the presence of a functioning repressor, right? Lac I is the opposite. It's inducible and positively regulated. So it is positively turned on, right? Because it's normally off, right? So that's an important distinction between those two as well. Okay, you have to, and it's hard to kind of sometimes get those things clear in your head, but you have to think about what is going on, right? If something is inducible, it needs an inducer. What's the inducer in the lac operon? No, that's the repressor. The inducer is lactose. Lac I is the repressor that binds to the lac O, the lac operator, in the absence of lactose or allolactose, depending on what you want to call it. Right, so these differences are important, and there are differences that are difficult to tease out without really understanding what's going on. Right, and that's not meant as a lecture, even though this is a lecture. Ha! But I'm just trying to get across how important it is that you understand those finer aspects of those two systems, because they essentially kind of tell us how those things work, right? They're not just like, you know, ancillary details. Okay, so allolactose is the inducer in the lac operon. Tryptophan tryptophan and TRPR are the repressor in the trip operon. Okay, so that's the big change in gene expression or regulation. What do we call it? There are a couple of different ways of calling it. What do we call it in the trip operon when we dial that back a little bit? There's a particular word. Begins with an A. Anyone remember what that's called? Not the big scale change, but the small scale tuning. That's right, attenuation. Hey, all of you got it. Right, so that's essentially kind of adjusting the level of output of the trip operon in response to levels of tryptophan, right? Not high enough to turn it off, but not low enough to allow it to be completely on either. Okay, so how does that work? What part of the trip operon is needed for attenuation? Has a particular name. It's not the TRPO, that's where the TRPR binds to. So operators are typically where repressors bind, right? It's the leader sequence, right? So essentially what happens here is that TRPO is no longer an issue, right? Because transcription has already started. So TRPO is unoccupied, right? Otherwise that TRPR tryptophan repressor would block transcription entirely. So transcription has started, and now we have a small upstream region. It's actually called it's called a 5' UTR, but I don't think that's a really good word for it because it is translated, right? That's the whole point. It's called the leader sequence, right? So what region is really important? There are 4 of them in the leader sequence in terms of detecting tryptophan levels. Let's see, so now I'm going to be drilling down into the details because you have to understand this stuff to get the bigger picture. Which region? 1 through 4. 25% chance of getting it right if you guess it. Nope. Which region is important in detecting tryptophan levels in the leader sequence? And it's region 1. All right, not the promoter. No, the promoter is way further upstream. That's already done its job, right? We already have transcription. Transcription is occurring. What the leader sequence determines is whether or not transcription continues, right? And so in region 1, as Peyton Davis said, there are 2 codons for, guess which amino acid? Oh, not methionine. That's in every protein. What does the T stand for in Peyton's response? What's the operon that we're working with? Tryptophan, right. So essentially in that region 1, you have 2 codons that needs the presence of the output of the trip operon, right? So what happens if tryptophan is present at a high enough level so that those so translation can proceed through those 2 trip operons? What happens then? So again, there's enough tryptophan around that translation can proceed, but termination transcription, yes, but how? How does that work? Right? Again, enough trip that translation can proceed, but not enough to repress the trip operon as a whole. How does that termination work? Right, region 3 pairs with region 4 because where is that ribosome now? Once region 4 is produced, where is that ribosome sitting? Remember, RNA polymerase and the ribosome are going along. Right, it's over region 2. And so it stops, so it basically stops region 2 and 3 from forming a loop, right? Because region 2 is covered, because the ribosome's there, we've got region 3, right? Can't form a loop. And so region 3 and 4 form a loop, and that's your terminator loop. Does that make sense? It's really important that it does. Right? And that's purely because there is enough tRNA with tryptophan that translation is not paused in region 1. Right? So that ribosome continues through, and at the same time, the RNA polymerase keeps transcribing the leader sequence so that when the ribosome is on region 2, region 3 and 4 have been produced. So they form a terminator loop and thunk, kicks off RNA polymerase. Now, what happens if there is not enough tryptophan tRNA for translation to continue? What happens then? So essentially we're saying there's not enough tryptophan in the cell. We still need the trip operon to be expressed. No termination, but how? Right? So your ribosome is going to pause on region 1, right? Because that's where the trip operons are. So what forms now? An anti-terminator loop between loops regions 2 and 3. Right? So remember, this whole process is coordinated, right? We've got our RNA polymerase going along. No worries. We've got our ribosome going along, right? So if the ribosome is paused on region 1 because there's not enough tryptophan in the cell for tRNAs to come in and it continue translation, then the RNA polymerase wouldn't have got as far as transcribing region 4, right? So it's still in the business of transcribing region 4, but 2 and 3 have already been produced. So those 2 and 3 produce a loop. And so when RNA polymerase continues on and makes region 4, here's region 3. It's already in a loop. It can't form a loop with region 4 because it's already paired. And because it can't form a loop with region 4, that termination loop or terminator loop isn't formed, right? So now the RNA polymerase goes, sweet, and it continues on and it transcribes the rest of the trip operon, like the structural genes, the stuff that matters. Right, so it's really key how those two things go together, right? Transcription and translation, because you're using the translation aspect to control transcription. And the way it does that is by what loops are formed, which depends on whether or not tryptophan is present at sufficient quantities for there to be enough tRNA with it on for translation to occur. How about that? Does that make more sense now? If it doesn't, ask, and I'll think of a different way of explaining it. Yeah, slide 25 is a really good one, to be honest, but sometimes it just helps to work through things. Yeah, this one. Gotcha. It helps to work through things and also to look at things from a different angle, right? So I think we taught this from the perspective of, not there being insufficient tryptophan, so this is what happens, and then you can work it at the other direction as well, right? Okay, what about lactose? Okay, what's the output of the expression of the lac operon? What metabolite will increase in concentration that the bacteria cares about? Glucose, right? Can bacteria or their gene expression mechanisms detect glucose directly? No. And it can't. And it's actually surprising, right? Because you'd think glucose would be a really important thing to be able to directly measure. No, don't worry about it. But we can't. Even eukaryotes can't, right? You yourself, your cells are unable to detect glucose levels. So we use a proxy, right? So what goes down, yeah, there you go, cyclic AMP. And that goes down as glucose levels go up. So it's like this teeter-totter, right? Glucose goes up, cyclic AMP goes down. Glucose gets consumed, right, faster than the galactosidase is producing it from glucose. Yes, an inverse relationship, right? So as glucose goes down, cyclic AMP goes up. Now, how how do we translate cyclic AMP levels into the fine control of the lac operon? What protein do we need? CAP, what does that stand for? P is for protein, that's easy. There you go, catabolite activator protein, right? And so what consequence does cyclic AMP bound to CAP have when it comes to the lac operon? You know, what does it do? Yeah, it doesn't so much turn it on, it just makes it work better, right? So what turns it on is the lack of lac-I, right? Which is removed when lactose is present, right? So that's the big scale on, off. Yeah. CAP with CAMP does exactly what Peyton and Angela says. It increases the efficiency of transcription, right? It promotes RNA polymerase binding, stability, right? Because it typically wants to fall off, right? So it's going to work a lot better, right? So now we've got, you know, we're going to have gangbusters amounts of transcription, right? Because it's going to work very efficiently. We're going to have more lactose permease, right? So more lactose comes in. We're going to have more beta-galactosidase, so more of that gets converted into glucose and galactose. Now our glucose levels start going up. What happens to cyclic AMP? If glucose levels go up that way. CAMP goes down. CAMP goes down, there's less CAP-CAMP, right? And so the lac operon functions less efficiently. So there's less beta-galactosidase produced, there's less glucose produced, so then CAP goes back up again, and we get more expression, right? So it's essentially kind of maintaining it like homeostasis, right? Temperature or blood sugar or whatever, right? It's a way of maintaining levels just where you're at, right? But the lac-I repressor means that if no lactose is present, there's no point transcribing any of this, right? It's not a nutrient that is always found, right? So lac-I shuts off completely the lac operon. Doesn't matter whether glucose levels are high or low, right? If lactose is not present, you don't get expression of the lac operon, right? So that's why it's a positively regulated inducible system, right? You have to tell it to come on. Tryptophan is always required, and you have to tell it to go off when you don't want any more of it. How's that help? Cool. So again, remember what's the purpose, right? What's the bigger context, right? Is this an essential metabolite or thing, which tryptophan is, or is it something which only sometimes may be present, which lactose is, right? You can think it's quite obvious if something is only occasionally present in your environment, you only turn on the system that processes it when it is present, right? So you want it to be off most of the time. Right, saves energy, you know, saves, you know, ribosomes have to do all, you know, they have to translate everything, right? So if you're clogging up the works with proteins being made that you don't need, you're not going to be making other proteins, right? Whereas tryptophan is an essential metabolite, right? No tryptophan, no translation, right, of any protein, because almost all proteins, not all of them, most of them maybe, have tryptophan in them, right? So the issue here is that, well, we always need this, but we don't always need a lot of it, right? So now it's a case of it's got to be always on, but then we dial it back or just turn it off completely when it's not needed, right, in response to varying levels. Okay, so that's essentially like the two halves of control of gene expression in prokaryotes, right? It's very much context. specific. Does anybody have any questions on prokaryotes before we move on to eukaryotes? Nope. Awesome. Well, if any do occur to you, just give a shout. So, question one, I should have reorganized it in the meantime. Been meaning to mix this around a little bit. So my guess in terms of control of transcription in eukaryotes, it's an issue of what do all these different things do? Is that right? That's just a guess, but that would be the guess that I would make. Or is it the specific roles of certain components or aspects of it? Where do you want me to focus? Specific roles. Okay, so I'm just going to invigorate myself. So essentially, if we want to draw out a gene, a story, right? So really, most of the, well, not all of it actually, a lot of the action in terms of gene expression is at the level of transcription. It's not only, obviously, there's epigenetics and, you know, all the stuff that comes after. Now, let me get some different colors. All the stuff that comes after transcription. But transcription is obviously super important. Where the hell my red go? Ah, there it is. Hiding. So when it comes to eukaryotes, there's a high degree of modularity when it comes to these regions. And so all of these regions, and it's really important to state this clearly, are DNA sequences, right, that exist in the upstream region, right? And those DNA sequences are recognized by proteins. Really, that's the root of all of it, right? So depending on two things, which sequences are present and which proteins are present in that cell determines whether or not we get expression from this particular promoter, right? So keep that in mind. It's basically it's a interaction between the two, right? The DNA is going to be the same in every cell, right? But which sequences are upstream of a specific gene allows that gene to be turned on or off in specific cells based on what proteins are present. And so we have a whole bunch of different regions. First up, I'm just going to get this one out of the way. We have, oh yeah, sure. Sorry, just got to get rid of this reminder. Snooze. Right, we have insulators, right? And really their job is simply to stop any proteins that bind here from affecting either positively or negatively any genes that away, right? Off of the whiteboard. So right around where my ear is, let's say, right? Because DNA can bend in all kinds of directions. And a lot of these work by bending DNA. So if you want to make sure that only this gene is affected by these particular proteins, you need to have an insulator to basically wall off that gene from anything else. right? So you don't have inadvertent gene expression from genes which you don't want to be on, right? That's a fairly simple kind of concept to get. Does that make sense? Yeah, so it protects upstream, exactly. Because, I mean, I can't write on my face, right? So if I was to write like another gene on my face here, this whole region would be the regulatory region for this gene. Sorry, angle a little bit, I'm getting wind blown into my mic, right? So we don't want anything here to activate gene expression of this gene go in this direction, right? So we basically block off that that gene by making kind of in a way, you know, like building a little wall between those two genes. They share the same chromosome, but you don't want them to be coordinately regulated, right? They are going to be expressed under different contexts. So you need to insulate them from each other. Cool. Okay. So then we have, pardon me, a whole bunch of different regions, right? And these are split into regions which can bind transcription factors, regions which bind activator proteins, and regions which bind silencer proteins, right? And so this is almost like a toolkit. So let's say this is a heat shock. If I can write that, protein. Right? That coding sequence over there is going to encode a protein that helps respond to heat shock. Now you may have you know, a couple of different transcription factor binding sites. You may have some enhancers. And you may also have silencers, right? This would be, each combination of all of these is going to be largely unique for each gene. So, for example, under, let's take a picture of this, and I'll post these up afterwards. So let's say right, we have you're out in the desert of San Antonio. TF is transcription factors. Sorry. I'll get that in the next picture. Transcription factors are what help recruit and stabilize RNA polymerase. So they're part of the initial process of holoenzyme formation. Okay, so you know you've been downtown, you haven't got a hat on, you know you haven't been inside much, and you're getting super hot. Now you're going to have some transcription factors come in. they're going to bind to your response elements in the promoter. We're going to, crap, I wish I had a different color now. Oh well. So here's our heat shock transcription factors. HS stands for heat shock. Let's make sure I got this at a right angle. Now, RNA polymerase assembles on that promoter. And we need, so the transcription factors facilitate that. And now we also need these are TAPs. So these are transcription activator. I'm going to get rid of this because we don't need this. Proteins, right? And now these, via mediator or co-transcriptional activators or other proteins that are part of this complex, these will activate transcription. So you need both the transcription factors, which bind to these response elements in the core promoter. They recruit RNA polymerase, and you also need our enhancers to be bound by transcriptional activator proteins to activate transcription, right? Those two things together get you transcription. How's that work? I'm going to hold this as best as I can so you can see all of it. Any questions? Okay, so under a different context, I'm just going to take a picture of that and rub some stuff off. Yeah, this is going to be more work than I thought. Come on. Yeah, that'll do. So now if it is cloudy, which doesn't happen very often in San Antonio, that's my best. Maybe we've got some lightning, get some good thunderstorms here, right? So we don't have need for heat shock protein expression because we're not overheating, right? We don't have the conditions necessary for expression of that. And don't forget, all of these heat shock proteins, and there are many, pardon me, will have a similar collection, not the same, but similar collection of transcription factor binding sites, enhancers, and potentially silencers, right? So that when these conditions do occur, you can get that coordinated expression through the production of those heat shock factors, which turn on heat shock protein expression, right? So this is the way in which eukaryotes do coordinated expression, right? Prokaryotes is simple. They just stick all of these genes together into an operon and just control the whole thing. Eukaryotes don't do that. They do their coordination via the production of these or presence of these proteins. Okay, so now maybe we've got the odd heat shock factor. Ooh, let's see if I can write this. Right, you know, maybe we have RNA pol, And perhaps we have the odd tap, right? But we don't want this to be expressed. So now, if we have, under these conditions, we're likely to have silencers present, right? And so now these bind to these silencer regions, right? So these are our silencers. They're also called repressors as well, like the ones that you learned about in the with LAC-I and TRIP-R. But now you can see maybe this does come in, right, and binds. It can happen, but our repressors, actually very much in the same way as in bacteria, they are bound to these sequences, and in doing so, they block the binding of, say, RNA polymerase, right, because it physically takes up space, or our transcription factors, or our transcriptional activator proteins, right? So now, even if RNA polymerase were able to assemble on the core promoter, right? Maybe this enhancer, this silencer isn't here, maybe it's somewhere else. You wouldn't be able to have the other proteins necessarily bind to that promoter, and you wouldn't have gene expression. Or if you do, you're going to have it at a much lower level. Maybe you can bind one transcriptional activator protein, so you get some transcription, but you can't bind both or three or four or whatever, right? Does that make sense? Yeah. So it's very much which genes get expressed depends on the combination of all of these things and their relative levels, right? You may have a low level of repressors or silencers. So much of the time you do get expression because these binding sites are not blocked. But some of the times you don't, right? You may have no repressors, in which case you will get expression, but only if these things are present. If they're not, you still don't get expression, right? So really the output of this gene in terms of transcription depends on the specific combination of all of these things, right? It doesn't matter if you don't have repressors present, if you don't have activators or transcription factors that will guide RNA polymerase in, right? It's the whole picture. And those all come together to control transcription. And obviously, the insulators over here block any of this information from activating expression of the gene further upstream. But again, just to complicate things, and this is where I wish I had a different color, right? This might be tap number 2, or there may be other regions in here, transcription factor, that's not HSF, right? Where other transcription factors can bind, or other transcriptional activator proteins, or even other repressors may be able to bind. And so this protein, or this gene rather, can be transcribed under a range of different conditions, right? But it all depends on what proteins in terms of transcription factors, repressors, and activators are present. Yeah, this is exactly Cree modularity, like cis regulatory element or response element modularity, right? And so you can have genes which are expressed in eyes, let's say for butterflies, right? Eyes and wings, right? but you don't necessarily have the same transcription factors or activators present in both eyes and wings. So there may be different response elements which drives expression in eyes versus a different one which drives expression in wings. And depending on what combination of all of this that you have is what dictates where, when, and how much that gene is expressed. You know, what cells is it expressed in? You know, you're never going to get alcohol dehydrogenase, for example, right, which turns ethanol into something less toxic. You're not going to get it expressed in your eyeballs, right? Because, you know, short of like going swimming in a vodka bath, you're not going to get any alcohol in your eyeballs, right? So no eye-specific genes will have any of the activator or transcription factor binding sites for alcohol. Well, yeah, essentially, I'm kind of like muddling things a little bit now, but essentially, what combination of cis response elements you have here, or cis regulatory elements, is what decides the specific transcription factor of that gene. And so you can also see how easy it would be in kind of an evolutionary context to gain maybe this one, right? So before, this gene was only expressed under one particular context, and now through mutation or maybe a transposon jumping or who knows what, right? Now you have an extra enhancer site, and that now drives expression somewhere else where that gene was not normally expressed, right? So now you have a new trait which you didn't have before. And that's largely how they think butterflies got their wing spots, because many of the proteins are originally expressed in the eye alone in like ancestral butterflies. But somehow the gene for that eye pigment picked up a different CRE, which is responsible for expression of genes in the wing. And so now you get a pigment protein, right, which you get in your eyeballs, is now being expressed in your wings in addition to your eyes, right? So really, and that's also why understanding gene expression, let's make sure I'm far enough back, in eukaryotes is such a difficult and complex process, right? Because it is a sum of all of that. And so really, to understand it in its completeness, you need to understand all of that, right? And that's a very difficult thing to do. Any questions? Pardon me? All right, if any do kind of pop up between now and lab, just give me a shout in lab, that's fine. So let's meet again at 2, and I will go through any questions you have about the Eugene lab. And I've got Eugene up and loaded. I went through it just as a refresher again yesterday, so that, you know, I'm sure that I know what I'm doing. I do this, but I do it once a semester, so sometimes I just forget stuff. So I'll be able to go through each of the different steps where necessary of the final lab for the course. And that should help. Several of you have emailed me with questions about specific parts. That should also help answer those questions, right? Because they may have been easy to miss or perhaps not covered sufficiently in any of those guides. Cool. All right, so see you back here at 2 o'clock, 2 p.m. And have a nice lunch. I'll see you in a little bit.